Okay, good morning, everyone. See, I haven't scared off absolutely everyone, which is a good sign. There's like half of you here. That's good. Today is a double lecture. So what we'll do is I'll talk for about, I guess, an hour, 50 minutes, depending on where's a reasonable break point. Then we'll have a little bit of a break in the middle for 10 minutes or so, depending on how time's doing. And then we'll come back again. So don't worry, I'm not just going to talk at you for 2 hours straight. Today, we're going to be talking about our first models of evolution. We talked about principles of evolution a little bit and some misconceptions on Monday. Today, we're going to be thinking about how do we actually start constructing some models to help us understand evolutionary principles. We're going to start off with this first part of the course is about an area of mathematical evolution called population genetics. And these models of population genetics, they're all about really trying to understand how sort of key genetic related phenomena, things like dominance, epistasis. You might not be familiar with these terms, and that's totally fine. Dominance, epistasis, mutation, drift, how these and selection affect changes in genetic variation, how they change how they affect sort of the dynamics that we might see either at the genetic level or even sometimes at the phenotypic level as a result of those genes. Really, a lot of the time we're thinking about what we would refer to as major gene effects. So things that typically have a fairly large impact on phenotypes. So a quick bit of terminology. In fact, today there's going to be quite a lot of terminology if you're not familiar with biology. So today is particularly terminology heavy just because we're introducing lots of different biology terms. A genotype is the genetic makeup of an individual, whereas its phenotype is its characteristics. So, for example, you might have genes that give you brown or blue eyes, say, but that sort of brown or blue eye is your phenotype, whereas the actual genes that correspond to that are would be part of your genetic makeup, and so would constitute your genotype. So there's a slight difference in terms of also how we might model things. Anyway, a lot of traits are determined by these major gene effects. Okay, so we're going to be talking about that today and in the next few lectures as well. Later in the course, we're going to be thinking about phenotypic or quantitative genetics as well. So these you can think of it being generally as more like continuous traits, or really traits that are approximated as being continuous often. They might be controlled by genes with many small effects. They might also actually genuinely be continuous, like something like your height, which is both determined by your genes and the environment as well. But often we can also model certain other traits that are controlled by many different genes as being roughly continuous. So we'll be talking about those types of models a little bit later in the course. In terms of today, we've got quite a lot of different concepts to go through. Like I said, today is quite terminology heavy, so kind of bear with me. If you forget what any of the terminology means, just ask me, I'm happy to clarify. So we're going to be talking about Mendelian genetics. So Gregor Mendel, who's familiar with Gregor Mendel and his experiments with peas? A few people. Okay, great. So Gregor Mendel is a Bohemian monk who uncovered some of the fundamental rules of genetics. It was unappreciated at the time. And this was then later folded into an evolutionary synthesis with some mathematics during the early 20th century. So we're going to be talking about Mendelian genetics, what that means for ultimately different phenotypic outcomes that we might care about. We talk about alleles a lot. So alleles are essentially just genetic variants at a particular genetic locus, or you can think of it as a position in your genome. It could be a collection of different nucleotides or molecules within your DNA, or it could be just one single nucleotide. I mentioned genotype and phenotype already. We'll be talking about absolute and relative fitness. Fitness is a fundamental concept within evolutionary biology. So think of survival of the fittest. That fitness is related to that. We'll talk about selection coefficients, telling us when things are increasing in frequency or decreasing in frequency. Concept of fixation, which is when a particular allele is represented by 100% of individuals in the population, so everyone has that. And we'll also talk a little bit as well today about frequency-dependent selection. So that's when the strength of selection depends on the frequency of an allele in the population. So there are feedbacks within those, okay? So haploids. We're going to start off with the simplest models we can, and haploids are basically as simple as we can start. So haploids have just one copy of an allele at a particular location or genetic locus. So locus is just, think of it as a location within your genome. So one copy of an allele. There could be different versions of an allele that could exist at that genetic locus. Okay. So we have one complete set of chromosomes within each cell. And for humans, we have these in our gametes. So gametes are our sex cells, so ova and sperm. Eggs and sperm, they have just one copy of the chromosome. So when you're, we have diploid cells typically. So you have within each of your normal cells, your soma, you have one of your chromosomes will be inherited from one of each chromosome is inherited from your mother. One of each chromosome is inherited from your father. That makes you diploid. So you have two copies of each chromosome. That means that at each genetic locus, you have two alleles. But when we produce our eggs and sperm, we end up splitting up our chromosomes, and then just one of those chromosomes ends up in each egg or sperm cell. And then when we have fertilization, those two chromosomes one from the egg, one from the sperm fuse together, and then we go back to being diploid. So we start, most of the human life cycle in a sense is diploid, and then we go through this very brief haploid phase in terms of our gametes. Haploids are very common in the microbial world, especially bacteria. Most bacteria are haploid. They reproduce asexually. Those two things don't necessarily always go together, but they often do. There are also very unusual things in the natural world. I like to say that pretty much everything you can imagine somewhere, like every rule that you can think of is sometimes broken somewhere in some system in biology. So, for example, we tend to think of our sex being determined by whether we've got X and Y chromosomes. That's flipped around in birds. The order of it's flipped around. Also in things like Hymenoptera, so ants, wasps, bees, sex is determined by whether individuals are haploid or diploid. So they're called haplodiploids. Unfertilized eggs grow up into being males and they're haploid, and fertilized eggs, when they're laid by the female, by the queen, they end up growing up into being diploid females. So there's lots of weird stuff that goes on related to whether things are haploid and diploid. There's a bit of an old joke actually in evolutionary biology. When people say Jesus H Christ, they often say that the H is referring to the fact that Jesus was haploid because he was a virgin birth, a little bit similar to bees. So that's one that often comes up. Anyway, so we're going to be talking a lot about haploids and diploids, but today we're going to be focusing on haploids. Okay, so let's consider the simplest population that we can. Okay, so we're just going to have two types of individuals in this haploid population. Okay, so we just have individuals that are either capital A or little a. Okay, and this is going to refer to their alleles, and in this case, it happens to be also referring to their genotypes, because those two things are the same. We've only got one genetic locus with two possible alleles at that locus. So individuals can either be a capital A or they can be a little a. You can think of this as being a label. So we're going to make a few assumptions to make our model simple. We're going to assume that our population is asexual, so we don't have to worry about mating. We're going to assume that our population is very, very large. In fact, we're going to assume that it's infinite. This allows us to treat things as being deterministic, so we don't have to think about stochastic effects. In finite populations, really, we should be thinking about those stochastic effects or random sampling. That leads to things like genetic drift. We're not going to be thinking about that. We're going to essentially assume synchronicity, so that all births happen at the same time, all deaths happen at the same time. We're going to be starting off looking at discrete generations. So all births and all deaths being synchronized at the same time as each other helps with that. We're going to census our population or count it immediately after our births happen, and there's going to be no mutation or migration. So all of these things just essentially help us to simplify our model. Okay, so as our births and deaths occur synchronously, we can model time in discrete steps. We don't have to do, sorry, yeah?
So the last slide, sorry, I wasn't familiar. What is migration?
Migration, good question. Yeah, so migration would just be individuals coming into the population or leaving the population. So essentially, we're just assuming the population is closed. That turns out to be important when you're modeling something called gene flow, where the genetic makeup of another population might be different, and therefore it can kind of affect what the genetic makeup is going to be in the population that you're interested in. Okay, so we're assuming we've got these discrete steps or generations. We'll call these generation T. It's just going to be, this is time T, but it's a discrete step. And we're going to let N sub A, with that A being either capital A or little a, being the number of, or abundance of individuals with that particular allele, okay? So we're just tracking the number or abundance of those individuals corresponding to their alleles, or in this case, also their genotypes. And we're going to suppose that each individual produces B sub I offspring per generation. And then everyone has the probability of dying D sub I per generation as well. And all we want to do is we want to write down an equation that tells us, okay, we're in current generation T. What is the generation going to, what is going to be the population makeup in generation T plus 1? Okay. So really we want an equation that's N sub I. So our I is just going to be our allele, like capital A or little a. In generation T plus 1, what's this going to look like? Well, I'm going to start off with what we had before. So it was our generation, generation T. How many individuals did we have at allele capital A or little a? We're going to have births. So we're told that each individual produces BI offspring per generation. So we're going to start off with what we had before, 1, and then we're going to add B sub I individuals. And then we're also going to have deaths as well. So we're told that there's a probability of death for anyone of DI. So 1 minus DI, D sub I are going to survive. We've got our proportion that die, D sub I, a proportion of new offspring, B sub I, and then our original individuals. Yeah.
Here it says that like the genotype I produces BI offspring, so it's like the counts, not the rate, but here we're multiplying by n, so we're treating it as the rate.
Well, because we're doing these discrete generations, you can think of this as a reproduction factor as opposed to a rate. Sometimes it depends on the textbook that you look at. Sometimes people refer to these as rates. Sometimes people refer to them as like reproduction factors. So each individual is producing this number of offspring in a given time step. It does vary a little bit between textbooks, though. I'm not going to be too picky if you were to refer to this as a rate, because it does differ a little bit based on how people feel about it. Yeah.
So in constructing this model, why did we account for like 1? Like, why didn't we just do BI times DI times NN?
So we've got this 1 in here because we're assuming in this particular model that we have overlapping generations. So individuals don't die. So for example, if we had, say, annual plants, where plants only exist for, say, the summer season, they produce all their seeds, they all die in the winter, and then the next lot of plants come up, then we wouldn't have this 1 here. Mm-hmm. This is just saying that all the adults don't die instantly. Yeah, but there's a good question. It's just a slightly different type of model. Yeah. Yes?
I don't know why we have a term DI times BI is going.
Yeah, so we're assuming here that if you think of this as like, these are the individuals that are born and the individuals that have survived from the last time step. And then we're told here that all individuals of genotype I have probability of dying. So think of this as all of the individuals that we have, and then we're just going to apply this factor here as the ones that then survive.
So we are assuming that this can happen for all the newborns?
Exactly, yeah. Again, very simplified population, we're assuming there's no difference in the probability of death for juveniles versus adults. That often is not the case, but for the sake of simplicity and starting off, that's what we're assuming here. Okay, so this is all in terms of numbers or abundance of individuals. Typically, a lot of population genetics models, they focus on the frequencies or proportions of alleles and genotypes in a population. So we can very easily convert our model that we've got above. Oh, in fact, I missed a slight line out here. Let me just take a step back just a moment. I'm going to just define this here. The bit I'm going to highlight in red, I'm going to define, sorry, as WI. So this whole factor out the front, I'm just going to define as WI. And this is my absolute fitness or reproductive. Okay, so we'll be talking a lot about fitnesses. This is going to be my absolute fitness of this genotype. Okay, now let me go. So now we're going to be talking about frequencies. So we can convert our abundance into a model with frequencies. Very straightforward because we know how many individuals there are of type capital A and type little a. Those are the only two types of individuals in the population. So we can just define some new terms. So let's define P to be equal to n sub A, capital A that is, divided by the total population size. These could have T's in them, but I've dropped the T's just for brevity here. Likewise, Q is going to be our proportion of individuals that have little a as the allele. And clearly this is just equal to 1 minus P because there are only two types in the population. These proportions, P plus Q, have to equal 1. Nice thing about doing this is because they have to equal 1, if you know what P is, you know what Q is. So that means we don't have to, here essentially we've got two equations for the population if we care about their abundances. If we just care about their frequencies, we only need one equation ultimately, because we can work out what the frequency of the other one is as soon as we know one of them. Okay, so we had an equation for our abundance. Now we can turn that very simply into an equation for our frequency dynamics, and we just need one of these. So I'll just choose P. So this is our frequency of allele capital A in the population in the next generation. Well, what do we have? Well, here we just defined it as NA, so it's N capital A over the total population size. So this is just going to be N sub A, capital A at T plus 1, divided by the total population size in the next generation. These are just the equations that we had all the way back here. So I can just substitute this in with my absolute fitnesses. So we're going to have W sub capital A times by NA generation T. And then similarly on the bottom, same again. And then equivalently, W sub little a is the fitness of our other allele times by the number of individuals with that allele in generation T. So what I've done there is I've just substituted in the equations that we had for our abundance in the next generation. Okay, what do we have now? Well, if we use this slide here and divide throughout by the total population size, we're going to be able to convert this to frequencies. So if I divide top and bottom by our total population size, so N sub capital A at time T, likewise N sub little a at time T, if we add those two things together, divide throughout by that, we're not going to be changing this fraction at all, but we'll be turning it into something in terms of our frequencies. So now we're going to have W sub capital A times by P of T, our frequency of allele capital A. And then on the bottom, we'll have W sub A times by PT plus W sub little a times by Q of T. And then since P plus Q is equal to 1, we can eliminate Q and replace it with 1 minus P. Now, obviously, this doesn't have all of the information that we had in our abundance. We've reduced those two equations down to one equation here. If you only care about the frequency dynamics, then this is all that matters. It doesn't tell you actually how many individuals are in the population because we no longer have that information in this equation. So if you wanted to have all the original information you had, you would also need to have an equation describing, say, the change in the total abundance of individuals in the population. But we're not going to care about that. So that doesn't really matter. Okay. So we've got an equation now. This is a difference equation that describes the change in the frequency of our capital A allele. And recall this, these alleles, they could just correspond to any trait that we might care about. Think about something like eye color. You know, suppose, I mean, we're talking about haploids here, but just for argument's sake, let's just think of something like eye color, just for simplicity, as something that we can relate to. So suppose this was blue eyes and brown eyes. Obviously, we're diploid, so it's not quite the same, but think of it as just a trait difference between these two types of individuals. Okay, this is describing how those traits change over time between different generations, depending on the absolute fitness of those different types. Okay, so we can simplify things even further by talking about the concept of relative fitness. So these fitnesses above, these W sub A, either capital A or little a, they are the fitnesses of those different types. It's telling us something about how much they're able to grow, how quickly they're growing. We can think about the relative fitness. In this case, we'll define V sub capital A is equal to W sub capital A divided by W sub little a as the relative fitness of allele A, capital A. The problem with talking about these models often is it's convention to use capital letters and little letters, but talking about them becomes a little bit harder if you want to just say A. Okay. So we can do a little bit more simplification here. If we just divide through by W sub little a, we'll end up with P in generation T plus 1 is equal to what we had previously, but divided by W sub little a everywhere. And then that very nicely cancels down. Just have one parameter V sub A. We have V sub A times by P of T divided by V sub A times by P of T plus 1 minus P of T. And if you want, you can gather those P of T terms together on the bottom. You have P of T times by 1 plus V sub A plus 1. So this is a difference. Yep.
Should the top A on your first equation be a lowercase a? Sorry, in the denominator, your second term.
Oh, yes, thank you.
Yeah.
Yeah. Thank you for spotting that. Yeah, that should be a sub little a. Thanks for spotting that. Okay. So we've got this is just a difference equation. Yeah. Do you have a question?
Oh, no.
Okay. So this is just a difference equation. Who's worked with difference equations before? Yeah, most people. Okay, good. So if you've only worked with differential equations, these are very similar, but they're in discrete time. We have a term, V sub A, which is our relative fitness of our allele capital A. And this equation tells us from one generation to the next, how do we expect the frequency of that allele to change? Now, intuitively, we'd hope that if our allele capital A has a higher fitness, it has a higher relative fitness, so this is greater than 1, it's V sub A, then it should increase in frequency, and it should decrease in frequency if it is less fit, right? That's kind of what we understand by concept of fitness. Things that are fitter should perform better, so they should spread within a population over time, and things that are less fit should decrease in frequency. So a little question here for you guys. So suppose the absolute fitness of each genotype. so our W sub A's, is multiplied by some same factor. Let's call this X in generation T. So this is some time varying thing. And this could represent variation in, so right, is that the BC alert that's going to hit everyone this morning I heard about? Yeah, so this could represent some variation in resource availability. You know, resources might vary between seasons or over a long period of time. There could be population density effects on survival as well that affect both of our genotypes the same. What we're going to know is how does this affect the equation above, and ultimately how does this affect selection in terms of these alleles? Any ideas? So if we're multiplying both of our fitnesses by the same thing. Yeah.
Remains the same.
Exactly. So what this tells us is, let's see if I just do it explicitly here, we're multiplying the absolute fitness of each of our alleles by the same thing. Obviously, this just cancels out and we end up back in the same model. So as long as environmental effects affect both of the alleles the same, then everything just factors out and they don't affect selection at all. It's kind of like a quite, you know, profound thing. It means that we don't have to worry about mapping all of those environmental effects. We only have to care about their relative fitness here. Yes.
All those conditions earlier when we were coming up with the equations, were those
Yes.
be accounted for in the x of t then, or is it just a separate model?
That's a good question. It's that would be a different model. Yeah. So these kind of things I'm thinking more about, yeah, there's some kind of variation in the environment in terms of like resource availability, you know, good seasons, bad seasons, but everyone is equally affected. It wouldn't apply, for example, if one of these alleles performs better in bad seasons, one of them performs better in good seasons, right? Because then those effects wouldn't be the same. We would say have an X of T, and let's just change this, say, to a Y of T, right? And therefore it would change our bliss B hat, right? That would affect it. But in terms of sort of just multiplying both of these fitnesses by the same thing, is suppose you suddenly flooded the environment with lots more nutrients and both genotypes could perform equally well. You know, you've doubled the amount of food, then ultimately this doesn't end up affecting anything if there's no differential effects. I mean, somewhat obvious, hopefully, but it's kind of worth pointing out that it does actually fall out of the model. It doesn't matter. Okay. So I mentioned this is a difference equation. It's also referred to as a recursion equation. So it follows that the change in allele frequency in a single generation is just given by the difference between what's in the next generation, what's in the current generation. It's by definition, right? So we can call this delta P. And this is just going to be the difference between what we have in the next generation and the current generation. Again, this is just a bit of algebraic manipulation, right? We have an equation for P of T plus 1 from the previous slide. In fact, I'm gonna go back to our absolute fitnesses rather than our relative fitness, just because there's sort of various reasons as we might want to work with one versus the other. But for simplicity, I'm going to stick with absolute fitness. So we had W sub capital A times by P of T divided by W sub capital A times by P of T plus W sub little a times by 1 minus P of T. And then we're going to subtract the frequency of this capital A allele in the current generation. And so this is just simple algebra, so we just need to now combine these fractions, turn P of T into a fraction. So what does this look like? Well, let's write it out explicitly just so everyone's on the same page. Have our W sub capital A times by P of T minus P of T times by everything that was on the denominator. We have a W sub A P of T plus W sub little a of 1 minus P of T, and then all over what we had before. just a little bit of algebraic manipulation here. We can gather our terms. We can expand this and then refactorize it if you like. So what are we going to have here? We're going to have W sub capital A times by P of T. So we've got something linear in P of T there. We're going to have a P of T squared here. We have another P of T squared here. And both of those P of T squareds are going to be multiplied by our absolute fitnesses. So that means that we can bring these absolute fitnesses out the front. We have W sub A, W sub capital A that is, minus W sub little a. Up front. Remember, both of those are going to be multiplied by a linear P of T. Linear one here, and we have this linear one out the front. And there's a quadratic term for each of these. So there's a P of T squared here and a P of T squared here for each of those absolute fitnesses. If this isn't immediately obvious, that's fine. You can always work through this in your own time just to make sure. But essentially all we're doing is just expanding this out and then refactorizing it. And the denominator doesn't change. Why might we want to write this in this way? Well, it turns out we're going to talk about something called selection coefficients in a moment. But we can see straight away that here, the difference in these absolute fitnesses is going to matter, right? If ultimately this is the change in frequency of allele A. And note that the bottom can't be negative. The P of T and 1 minus P of T also can't be negative. So the sign of delta P is just going to depend on the sign of W sub capital A minus W sub little a. So the difference in the absolute fitnesses. So again, that should intuitively, we talked about, okay, if an allele is has higher fitness than another one, then it should increase in frequency, and vice versa, it should decrease in frequency. Okay, so that will only happen depending on their relative fitnesses or absolute fitnesses here. Okay. One of the important terms in population genetics is a selection coefficient. And these selection coefficients are essentially just, a lot of this is rewriting the equations into some slightly easier formats. So sometimes it's easier to work with relative fitness, sometimes it's easier to work with absolute fitness, and sometimes it's easier to work with selection coefficients. So these are all just slightly different ways of structuring the same model. And we can define our selection coefficient S sub D. We're going to define this here for our capital A allele. Going to be the difference in their fitnesses divided by the fitness of allele W sub little a. Allele little a, so W sub little a is on the bottom. This also means we can rearrange this for our fitness for our capital A allele in terms of the selection coefficient, 1 plus SD times by the fitness of our little a allele. Okay. Might be thinking, why do we want to do that? Well, just go back up here for a second. Notice that we have a difference in fitnesses as appearing here and here. We also have these fitnesses appearing in the denominator, kind of like with our relative fitness model, where we were able to eliminate our two fitnesses, W sub A, capital A and little a, that is, and replacing them with just one term. We can do the same thing in terms of a selection differential. Like I said, this is just a rewriting these equations in a slightly different format. And depending on the kind of thing you're doing, sometimes it's easier to work with one as opposed to another. So our delta P, so our change in the frequency of allele A in one generation, we can write this in terms of S sub D now. How do we do that? Well, we note that up here, we can replace the W sub capital A minus W sub little a with W sub little a times by SD. And then the rest of what we had on the numerator stays the same. So P of T times by 1 minus P of T. And then on the bottom, well, we had each of our individual fitnesses. So I'm going to replace that W sub capital A with W sub little a times by 1 plus S D. That's just using this rearranged form here. And I'm going to leave the W sub little a that we had there before times by 1 minus P of T. So this is nice now because we've got a W sub little a everywhere. So those are going to all cancel out. On the bottom, we're going to have 1 plus SD times by, oh, sorry, I've missed a P of T out here. Should be a P of T there. So in this first term in the denominator, there should be a P of T. So we're going to have a 1 plus SD times by P of T plus 1 minus P of T. So that means on the bottom, this is just going to cancel down to being 1 plus SD, our selection coefficient, times by P of T. And on the top, we have our selection coefficient times by P of T times by 1 minus P of T. And again, we've gone from having these two parameters just down to one parameter. In fact, I refer to them as parameters. These can actually be variables as well. They might change over time. How's the speed so far? Too fast, too slow, just right? Just right? Okay. Just say, I'm happy to always provide clarifications if I do start going too fast. If I'm not getting clarifications, I'm going to assume that everything is going at the right pace. So I will leave that to feedback from you guys. If I'm going too fast, please let me know. Likewise, you can heckle me if I go too slowly. That's also fine, within reason. Okay. So why might we care about this selection coefficient? What does it actually tell you about the fitness of the two alleles? How would you sort of, if you were to try and put this into words, what is this SD telling you? Yeah?
Who's gonna like dominate?
Yeah. So it's telling you something about which one is going to be selected for, which is kind of in the name, right? That's what we would hope for. In terms of what it is, we can think of this as being the proportional difference between these two things in relation to our little a allele. So if I was to write this in words, I would say that SD is the proportional difference in fitness between the two alleles. But crucially, with respect to allele little a. I could have defined this in terms of W sub capital A, right? It would have been slightly different. Here we're talking about things relative to this little a allele. We'll go for about 10 more minutes or so, and then we'll have a break. Okay. So far we've focused on deriving these equations and just kind of algebraically manipulating them to write them in slightly different formats. And this is all about describing how the frequency of our allele capital A, which we're just arbitrarily assuming that we're interested in how this allele is changing. Obviously, we can just flip things around and talk about allele little a if we wanted to. We're thinking about changes in response to selection. We've not actually talked at all about our fitnesses, what they are. You know, we've just introduced these W sub capital A and W sub little a. We've talked about absolute fitness and relative fitness. We've not talked at all about what they might be. But if we recall, we defined our absolute fitness at the start for allele I to be 1 minus di, where di was our probability of dying. And for this particular population, as was asked earlier, we're going to assume that we've got overlapping generations. So we have our individuals that survived and our number of offspring are produced by those individuals, our reproductive factor for those individuals from the last generation. So this equation here for W sub I go all the way back to the beginning, is just what was highlighted in red here. Okay, that was all the way back from our original definition. If you remember on Monday, we talked about the survival of the fittest and how that survival aspect can be a little bit misleading. We think too much about survival, we just think about this first factor here, the 1 minus di, right? What's surviving to the next generation. But it's also fitness crucially depends on what's being reproduced as well. So there's no point surviving if you're not reproducing. Likewise, you might reproduce a lot, but if everyone dies straight away, it doesn't matter. Okay, so we do need to factor in both survival and reproduction. It also means that differences in fitnesses between different alleles and different genotypes ultimately in the population can come from differences in survivability, this is referred to as viability, and also reproduction or fertility. So, a very simple exercise for you guys. Given these values here for these two alleles, their birth factors and their death probabilities, which allele is fitter and what is the selection coefficient in terms of allele capital A? So yeah, have a go at working that out just for a few minutes. Okay. So this isn't a trick question. As simple as it looks. So fitness of our capital A allele is just going to be 1 minus d sub capital A. So just from above, that's a half. 1 minus half times by 1 plus b sub capital A. B sub capital A here is 1. So we're going to have a half times by 2, which is just equal to 1. And then for little a, we have 1 minus the probability of death, which here is a quarter. And that's going to be multiplied by 1 plus b sub little a. B sub little a is a half. So we're going to have 3/4 times by 3/2, 9/8. So which allele is fitter? Little a. Yeah. So little a is bigger than, or W sub little a is bigger than W sub big A. So allele A is fitter. And then our selection coefficient, just the difference between those two fitnesses. In this case, we're going to define it as being relative to W sub little a. We're going to have 1 minus 9/8 on the top divided by 9/8 on the bottom. We'll have minus an eighth over 9/8 or minus 1/9. In case I've made a mistake. Yeah. Also, we can note that our fitnesses here are always going to be positive as we've defined them in this model. Note that this is not always necessarily the case. This is just kind of on the assumptions that we've had so far. 2 seconds and I'll answer a question. So our selection coefficient is always going to be between -1 and infinity. as we've defined things here. Yes.
The question asks for like the selection coefficient for the big A allele, so we have to switch that around and do.
So it was it's defined the same way as we had here. So this, I appreciate, yeah, maybe that was slightly, maybe the wording of that maybe wasn't slightly clear, so I apologize. Yeah. So here I defined this as the selection coefficient. Yeah, maybe I should have said that here that this was for allele capital A relative to allele little a. And then the selection coefficient for allele A. Yeah, that's fair enough. This bit here was maybe a little bit ambiguous. So apologies if that was, if you wrote it the other way around. I mean, ultimately because this is symmetric, or in the sense structurally symmetric, you could have defined this as this selection coefficient in terms of W capital A instead. Obviously, you would have ended up with a slightly different answer. It would have been positive rather than negative. Oh no, sorry, you still would have been negative, but you would have ended up with a slightly different selection coefficient. Ultimately, yeah, when you would substitute this back into this equation here, you would end up with a slightly different equation. It wouldn't look quite like this because you would have defined it with a W sub capital A on the bottom instead. Ultimately, though, they do give you the same things. But yeah, totally reasonable point. It's good for me to know that because when I'm writing exam questions, I'll try to avoid that.
So should we stick with one, or depending on the problem, we should use one?
It depends. Like, there's no right answer to that. Sometimes it's easier just to be like, I'm always going to do it in terms of the thing that I think is positive. In this case, it turns out to be the one that's negative. Sometimes you maybe are focusing on a very common allele in a population, and you're thinking about whether a rare allele can invade, whether it's going to be selective when it's rare. So there might be some cases when you want to frame it in terms of the rare allele invading. You might want to think of it as being your selection coefficient being positive and it dying out as it being negative. Whereas if you frame it in the other way around, then your kind of concept of like thinking about something increasing or decreasing is going to be reversed. So there really isn't an absolute right or wrong way to do those. It kind of depends on the context. Honestly, if this was in an exam and you framed it the other way around, as long as your answer was ultimately consistent, you'd still get the marks for that kind of thing. So the main thing is just making sure that you're consistent, really. Okay. Any other questions? Okay, that seems like a pretty good time to break. So why don't we take like 10 minutes and then we can reconvene and go through the second half, okay? Right, so we've just gone through haploid models and talking about the frequency dynamics of these models. And we've talked about how we can rewrite them in different ways, and we can end up with ultimately this selection coefficient, which is beneficial in certain circumstances. For example, when we, some of the COVID work, so I talked about COVID the other day, some of the COVID work when say a new variant comes in, one thing that people would often try and classify as like a selection coefficient. They would talk about this kind of thing here. You could think of this as being maybe the W sub little a might be the dominant variant of COVID at the time. And then the W sub capital A would be a new variant of concern. Don't know if you remember like the alpha and beta and gamma and delta variants and Omicron, and all of those variants came up at different points. And so we, try to classify how, like, based on sort of some early dynamics, you can think of its reproduction as being like transmission. You can think of the things like recovery or death as being kind of like the death probability. So you could kind of work out fitness based on what's kind of going on. And then you might want to say, okay, well, do we think this is going to keep growing exponentially? Is it going to spread into the population? Is it going to take over? And so people talked a lot about selection coefficients in context of things like infectious diseases. So we've talked about those. We can talk a little bit now in this second half about a bit more about these fitnesses and when we have constant selection coefficients versus variable selection coefficients. So what we've talked about now essentially is a constant selection coefficient. We didn't have our S of D. By the way, the D here is just put up here, this D here. It's just because we're in discrete time, in case that wasn't clear. So we've talked about these constant selection coefficients. This wasn't a function of time. It's just assumed to be constant. And it should be pretty obvious that the change in one generation of our frequency of allele capital A is only going to be positive if the selection coefficient is positive, and vice versa. It will reduce in frequency when the selection coefficient is negative. That should be pretty obvious. That's intuitively and mathematically, it should hopefully be fairly obvious. So selection always favors the fitter allele. The question is, what happens to P, our frequency of allele A in the long term, depending on the sign? What's going to happen to P? Yes?
If it's positive, then P should tend to 1. If
Mhm.
it's negative, it should tend to 0.
Okay, great, good. I was hoping, no, I wasn't hoping. I'm glad that you didn't fall into the trap of it saying it goes off to infinity, because we're talking about frequencies here, right? So it's bounded between 0 and 1. That's the key point. So P bounded between 0 and 1. So if our selection coefficient is positive, then P of T is going to tend to 1 as T tends to infinity for, can we write that? For 0 less than P of T less than 1. I'm going to call it P0 actually. So our initial frequency, if it's between 0 and 1, then we're going to tend towards 1 if our selection coefficient is positive, and P of T is going to tend towards 0 as T tends towards infinity. Again, when our initial frequency is between 0 and 1. What happens if our initial frequency is 0 or 1?
It's stable, so it stays there.
It's stable, so it stays there. We haven't actually talked about stability. Maybe I'll leave that as an exercise at some point. There's questions in terms of, when we talk about any sort of dynamical systems, is something stable? It's an equilibrium or a steady state. We don't actually know whether it's stable or not from this. It depends, well, the stability ultimately depends on this selection coefficient, right? 1 is a stable state, 0 is not a stable state, but if you start at 0, you stay there indefinitely. Because if we go back up to our equation here, let's get all the way back up to this equation here, we'll do it. So if either P of T is equal to 0 or P of T is equal to 1, delta P is always equal to 0, regardless of what our selection differential is, our selection coefficient. So even if you've got a very, very strong selection coefficient, because we've got, actually, no, maybe I'll ask that as a question. Why doesn't it go anywhere? What assumption do you remember that we had right at the beginning? Something that happens in normal biological populations that gives us variation in our genetic material, but isn't captured in this model. And that assumption in this model therefore means that if you start at a frequency of 0, you stay there, or if you start at a frequency of 1, you stay there. The question might be a little bit too abstract.
Mutation or migration.
Bingo, well done, Suresh. Yes. So there's no mutation, no migration in this model. If we had mutation or migration in this model, then you might you would be able to move away from those states. But because we're assuming that there's no mutation or migration, if you start at 0 or you start at 1, that means that you've only got capital A allele or little a allele. There's no way of getting the other allele, so you stay there. So that is a limiting assumption of this model, right? But essentially, let's write it here. If P of 0 is either 0 or 1, so it's in set 0 or 1, then no change in P. Obvious. There's nothing to select on, nothing happens. I'm bored. Okay, So that was a constant selection. Let's make things a little bit more interesting now. So suppose instead that the fitness of an allele depends on its current frequency. So let's suppose that for our allele capital A, its fitness is going to be some function f of p. And for little a, we'll call it g of p. Now, I've slightly skipped over a step here. I said the fitness of an allele depends on its current frequency. I'm only going to use p here. I could have written this in terms of q, but obviously q is just 1 minus p, so this is a simple substitution. Okay, so I've written both of these functions in terms of p, but essentially because there are only 2 alleles in this population, it's the same thing in terms of depending on its current frequency. If you know the frequency of one of them, you know the frequency of both of them. So, why might this be the case? Why might you think of any scenarios why the frequency of an allele, or sorry, I realize my question here is wrong. Why might the fitness of an allele depend on its frequency? Yes?
There's more of them, they might die more often, or they work more things.
Yep. So depending on what else is around in the population, it might affect your fitness, is kind of what you're saying there, right? Yeah.
There might be like cooperation in the
Yeah.
society.
So cooperation is a good example. So cooperation, if we're going to talk about cooperation and cheating later in this course, but if allele capital A was a cooperator and allele little a was a cheater, the more cooperators are around, the more you would expect the growth rate to increase. The fitness of them will depend on the frequency. So cooperation, more generally, interactions with other individuals. Or organisms. Now, these interactions could be direct, they could be indirect. So a direct interaction might be something like you're cooperating together to do some hunting, something like that. Indirect might be something related to other organisms. So it could be something like parasitism or predation. Why might those be there? Well, we'll talk about this again much later in the course, but parasites, pathogens, you'd expect them to specialize on the most common types in the population. So it might be fitter if you're more different from the most common type. So your fitness might depend on the frequency of other individuals in the population. So you're not directly interacting with them, you have a shared enemy. Okay, and it might pay to be different from your shared, from what everyone else is in order to avoid them. There's also examples of this in terms of things like mimicry as well. Do you want to be like other individuals in the population? That might help protect you against predators or against parasites. Okay, so these are just very generic functions. Suppose, for example, that the fitness of our capital A allele is equal to 1 minus P. Remember, P is our frequency of our capital A allele. And conversely, we'll say the fitness of our little a allele is equal to P. So if we want to, we can work out our selection differential. I'll do this consistent with how I defined it previously. W sub A at the bottom. So we're going to have 1 minus P over P, which is just going to be 1 minus 2P over P. That's my selection differential, selection coefficient. So here we've got two possible initial conditions here. So suppose our initial value of P, so our initial frequency of the capital A allele is 1% or 0.01 as a proportion. So show what happens to the population. Likewise, what happens if our initial value of P is 0.99, so 99% of the individuals have capital A allele. And in general, what happens for any initial value of P between 0 and 1? So have a go at that. Work out what happens to P in the long term.
My intuition said it should tend to 1/2 in both cases. Simply a stable.
Okay. That's your intuition. Yeah, work it out. Have a little, I guess, yeah, work it out in terms of the selection differential just to make sure that your intuition is correct. Okay. Before I go on, just to clarify something I said earlier, I just want to make sure it's clear. We can write the selection differential in terms of each allele, but we should be dividing by the thing that appears second, right? So I could write the selection differential as this instead, just to make sure that it's clear. It's not just what we divide for changing. The thing that appears in the denominator should be the thing we're taking away on the numerator. Okay, so we can write it in a different format, but it's not just changing the denominator, just to make sure that's clear. Okay, so this particular question, so start with, like, what happens if our initial frequency of our capital A allele is 1%? So if P of 0 is equal to 0.01, then all we need to do is look at our selection differential. So again, one of the nice things about looking at the selection differential here is that we don't actually have to, if we want to work out what happens in the long term, P is bounded. We don't have to actually go all the way back to this equation here or one of the other forms to work out what happens. We can just look at our selection differential, right? So our selection differential here is going to be equal to 1 minus, funny, 1 minus 2 times by 0.01 over 0.01. This is clearly positive, so P is going to increase. And suppose we start off at 0.99. Then in this case, our selection differential is going to be 1 minus 2 times by 0.99 divided by 0.99, which is clearly going to be negative. Okay. The numerator here is negative, so P decreases. So we know that when we're close to 0, so when our frequency of the A allele is close to 0, it's going to increase in frequency. When it's close to 1, when it's almost every individual has this capital A allele, it's going to decrease in frequency. So in general, what happens for any initial value of P? So we can see that the population will evolve until our selection differential, our selection coefficient is equal to 0. Okay, it's positive for low values of P, it's negative for high values of P. It changes sign somewhere. The point at which it changes sign is where it's going to evolve towards. Okay, so what is it? Just clarify, this is where there is no selection coefficient. No selection coefficient because we've got no mutation in this model either, no genetic drift. We're in infinite populations. Then once we get there, we're going to stay there. So what we need to do here is we just need to find the value of our selection coefficient when it's equal to 0. So our selection coefficient is 1 minus 2p over p. We need that to be equal to 0. For this to be equal to 0, we just need p to be equal to 1/2. So conclusion then is for any initial condition between 0 and 1, remember, if we're at 0 or 1, we're just going to stay there because there's no genetic variation. Then P of t is going to tend towards 1/2 as t tends towards infinity. In other words, a biological interpretation would be both alleles eventually end up at equal frequencies. Okay. So I mentioned this a little bit the other day about structuring answers. Again, this is like hopefully a reasonably good example of that, of like setting out a logical order, describing what I'm doing as I do this, describing things in words, ending up with some conclusion here at the end. One key thing as well I'd like to highlight is this, or the difference between this line and this line. So what's in red I would describe as a mathematical conclusion, right? You should hopefully be quite familiar with this sort of conclusion, right? For any initial value of P satisfy this criteria, we have what happens in the limit as T tends towards infinity, a very mathematical answer. So if I would ask you for a mathematical answer, I would typically ask you for something like this. If I ask you for like a biological interpretation, I'm asking you for something more like this. So, you know, if you present, imagine, you know, for a non-specialist or relatively non-specialist audience, someone who doesn't have a mathematical background, say a biologist, if they ask you to model this population, this happens to me all the time in the sense of, I work with empirical biologists, I do models with them. If I give them an answer like this, they're going to say, I don't know what you're talking about. Like, where's the biology here? It's all math, right? Both alleles eventually end up at equal frequencies, very easy to understand. So recognizing the difference between a mathematical result versus a biological interpretation, they're both saying the same things, but one is written in a slightly different language. So again, this is all about making sure that, yeah, we can do the math, but can we interpret it in a biologically meaningful way for someone who maybe doesn't understand the math? So whenever you're asked to do that, always try and reinterpret what your mathematical result is in the context of the original biological question. Okay, that was a slight aside in terms of structuring and relation between mathematical and biological answers. In terms of this actual scenario here, this is an example of something called negative frequency dependent selection, usually abbreviated to NFBS. And this is often referred to as rare advantage. So when our capital A allele was at very low frequency, it increased. And when it was at very high frequency, it decreased. So there's a negative feedback. So Javad, you were right. In the long term, your intuition was good. Things are going to tend towards equal frequencies because when the capital A allele is rare, it's very advantageous. So it will increase. When it's very common, it's not advantageous. It's disadvantageous. It decreases in frequency. Things balance out. This is referred to as well as balancing selection. Why might this occur? Well, common thing, and again, we'll meet this later in the course, is when, say, antagonistic interactions like between hosts and pathogens or predators and prey, plants and herbivores, when they evolve together or co-evolve, we can see in the case of pathogens, they tend to infect the most common host phenotypes. Then selection will favor those that are rarer in the population. This can be quite important because it can lead to long-term fluctuations in these different allele frequencies. Here they happen to settle down, but there might be reasons as to why these things cycle over time. It also promotes diversity, right? Our final point here is not just one of these going to fixation. We've ended up with more diversity. So these kinds of processes, negative frequency dependent selection, can help us to explain some of the genetic and phenotypic diversity that we see in the real world. Okay, let's think of a slightly different example now. Again, we're still talking about frequency dependent selection. What about if we reverse things? So now we're going to have the fitness of our A allele is going to equal P, and our little A allele is going to be 1 minus P. So I won't give you the selection differential this time, or selection coefficient. I'll leave you to work that out. Have a go at essentially doing the same thing, working what happens in the long term. Okay, let's see how you guys got on. So very similar to what we had before. Now our selection differential is going to be P minus brackets 1 minus P over 1 minus P. So this is just 2P minus 1 over 1 minus P. Same process as before. What happens if our capital A allele is initially very rare. P is at 0.01. In this case, our selection differential give me 0.02 minus 1 over 1 minus 0.01, which is negative. Clearly negative because we've got something small minus something large here on the numerator. So P decreases. So when it's rare, the allele A, capital A, gets selected against. And conversely, you can probably guess what's going to happen. Our selection differential is 2 times by 0.99 minus 1 if it's very common, minus 1 minus 0.99. Okay, so it's positive, so P increases. We can do the same process as we did for the previous example. So we can find P such that our selection coefficient is equal to 0. If we do that, it implies that P is equal to 1/2 again. However, this is an unstable steady state. So I was describing to Javad just now. If we have, these are just difference equations. If you've ever done stability analysis of difference equations, we can end up with steady states. A steady state is basically just telling you that nothing is going to change if you're currently there. You can think of this as being like a hill or a valley or a hill in this order, and you've got a ball, and the ball is stable in both of these states, right? It stays at the bottom of the valley, or if it's perfectly balanced at the top of the hill, it stays there. In the case of the previous example, if you think of this as being P is equal to 1/2, here where the ball is currently, if you move a little bit away from it in the previous example, you're always going to get pushed back to the middle. It was balancing selection. If you move this one a little bit to the left or right, so you decrease P slightly off of 1/2, it's no longer finely balanced, it's just going to keep rolling away. Okay, so although they're both steady states in both of these versions of the model, P is equal to 1/2 is a steady state in the previous example, and in this one, the previous one was stable, whereas this one is unstable. So it's an unstable steady state. Ultimately, if P is greater than 1/2, our selection coefficient is positive. And if P is less than 1/2, our selection coefficient is negative. So in other words, whichever allele is more common, will go to fixation. Fixation, remember, just being that allele representing 100% of individuals in the population, frequency of 100%. Again, this is kind of that biological answer here, as opposed to a more mathematical answer above. That might not be too surprising here. We've reversed the fitness, the frequency dependence, we reversed it around. So before we had negative frequency dependence, in this example, we've got positive frequency dependent selection. When might this arise? For example, prey organisms often present certain colors or display warnings. So plants do this as well in terms of like berries, don't eat red berries, that kind of thing. Prey do this as well. You know, toads, for example, can display different colors, warning that they might be toxic to eat to predators. In that case, it's better off, it's harder for predators to remember lots of different colors, right? It's easier for them to just remember red is bad, kind of thing, or yellow is bad. Therefore, you don't, if you're the prey, you don't want to be different from other organisms in the population. It pays to be like everyone else in this case. And it doesn't matter, maybe, you know, arbitrarily like why red versus yellow. Might not be any reason why. It's just whatever is more common gets selected for. So we end up with positive frequency dependent selection. So here, either P is 0 or P is 1 is a stable state. And you go towards either of those states depending on which one you started closer to. Okay. Okay, the last little bit that we're going to talk about is about continuous time. So today we've been talking about discrete time models, so discrete generations. But what happens if our population doesn't have this synchronicity? It doesn't have like all its births and deaths happening at a regular time. What happens if things are more continuous, kind of like they are in human populations? Okay, so we're going to now think about a small interval of time, delta t, and we're going to define delta b as the number of births and delta d is the fraction of the population that dies within that time interval, delta t. Okay, and we can do that for each of our, it should really be a b sub I, delta b sub I. Yeah. So define up here the absolute fitness across that given interval, delta t. It's just going to be the proportion that survive. So 1 minus di delta t times by 1 plus bi delta t. I'm going to assume this delta t is relatively small so that we don't end up with more individuals dying than are possible. Okay. So it's going to be a relatively small window, and then we're going to just take the limit and see what happens as delta t goes to 0. Okay. So we've got our selection coefficient from before. So our selection coefficient modified now across this window delta t is just going to be taking our difference in the fitnesses with these delta t's incorporated. So this is just our selection differential that we had before, a selection coefficient, or incorporating this window delta t. And then we can just substitute everything in there. So this, we could substitute everything that is in green at the top. And if we do this, we can do a little bit of rearranging at the same time. Just because time's a little bit limited, I'll do all the rearranging as well at the same time. I'll leave it as an exercise if you want to do it, or you can just trust me. We're going to have ultimately the difference between our birth factor and our death probability for allele A, and similarly for allele little a. It's going to be multiplied by delta t. And then we're going to have something that is of order delta t squared. And then on the bottom, we're going to have 1 plus b sub little a minus d sub little a times by delta t minus, again, something order of delta t squared. I'm not going to care about these terms here. You can write them out, but we're going to be ultimately doing a first order approximation, then letting delta t tend towards 0. Because delta t is assumed to be relatively small, we don't care about these squared terms. Okay. So like I say, assuming this time interval is very short, delta t is much less than 1, we can just write down that first order approximation. Just for clarity, we drop the order of delta t squared terms. I'm going to have b sub capital A minus d sub capital A minus b sub little a minus d sub little a, that's by delta t, all over 1 plus b sub little a minus d sub little a times by delta t. All I've done is I've just dropped the order squared terms from the previous slides. You don't necessarily need to write that out again. Okay. Now we can think a little bit about our limits. What happens as delta t tends towards 0? Well, as delta t tends towards 0, actually let's divide throughout by delta t. So if we define SC, this is going to be in continuous time. If we define this to be the limit as delta t tends towards 0 of SD of delta t, so our discrete time selection coefficient over a window of size delta t divided by the length of that window delta t, this ends up just being that term in the numerator. Okay, so we have B subcapital A minus D subcapital A minus brackets B sub little a minus D sub little a. Okay, so we've got rid of this delta t here. As this delta t goes to 0, we're just going to have 1 on the denominator. So we just end up with this term here in brackets. Okay, we just end up with this here. It's nice and simple. We've got rid of all of our time dependence. It's our selection coefficient in continuous time now. Ultimately, we can think about this as well as related to before. Our allele capital A is going to be selected for depending on the relationship between its birth and death rates relative to the other allele. Okay, so it depends on both its birth and its death rates, not just one of those two things, just like we had before with our discrete time selection coefficient. Okay. Because we're in continuous time, ultimately, we want to have an instantaneous rate of change for our frequency of allele capital A. So in other words, we want to have a differential equation, dp dt, which is just going to be the limit of delta t tending towards 0 of our delta p over delta t. We worked out our delta p earlier, just the change in frequency in one generation. Now we're thinking about things over a time window delta t. So what's this going to be? Yeah, I'm still taking our limit. Leave the limit in there for now. So if we substitute in our equation for delta p, whilst also accounting for this window of delta t, it's ever so slightly different to what we had before. We had SD of delta t times by p of t times by 1 minus p of t over 1 plus SD of delta t times by p of t. This isn't complete yet, so we'll write something else in here. This here is just our delta p from earlier, but with our arbitrary window delta t incorporated into our selection coefficient. That's all that's changed. One thing that's missing from here is this delta t here, so we need to divide by delta t. I'm just going to put this here. So our selection coefficient for an arbitrary time window delta t introduced into our equation for delta p, which is our change in one generation. Now it's a change in an arbitrary time window of the frequency of allele capital A. We're dividing through this delta t here because ultimately we're going to be taking a limit and we want to find a differential equation to give us an instantaneous rate of change for our frequency of allele A. So what happens here? Well, we worked out what this is, this limit here. And we defined this to be S of C. So our selection coefficient in continuous time is SC here. So that means dp dt is just going to equal S of C times by, well, we have P times by 1 minus P on the top. We can drop the T's here because we're working with a differential equation now. We know that P is time dependent, so we don't have to write P of T everywhere. You can if you really want to. And on the bottom, what do we have? Well, our selection coefficient with respect to delta t is going to tend towards 0. What we have as a little aside, S of D delta t is going to tend towards 0 as delta t tends towards 0. Okay, why does that matter? The amount of selection that goes on as this window tends towards 0. This discrete window is going to tend towards 0. That means that on the bottom, this denominator here, we're going to have 1 plus something tending towards 0 times by P of t. I realize I've got an extra bracket in here. Apologies. That bracket there should not exist. That should be on the outside. So I had a bracket here. This parenthesis should go on the outside here. So this SD of delta t was multiplied by the P of t. Apologies for that. That ultimately means that when this SD of delta t goes to 0, this term here multiplied by the P of t also goes to 0. So this bracket was in the wrong place before. That goes to 0, so we just end up with 1 on the denominator. So this is it, our equation for our instantaneous rate of change of allele A in continuous time. Okay. That is about all I've got. There are a couple of questions there. I might leave those as an exercise. I might actually just drop them and leave them for problem sheets in the future. But we can ultimately think about the rate of evolutionary change being maximized by thinking about what happens here. In fact, I will go through this briefly because it's kind of interesting. This will only take a minute or two. If we think about this right-hand side here, if I define this to be f of p, just some function of p, the rate of evolutionary change is going to be maximized when that right-hand side, the derivative of that right-hand side is equal to 0. Okay, so this is my instantaneous rate of change. My selection coefficient times by p times by 1 minus p. If I want the rate of evolutionary change to be maximized, then it's going to be maximized when the derivative of that right-hand side is 0. If I take the derivative of that right-hand side, let's do it here, df dp is just going to be our selection coefficient in continuous time times by 1 minus 2p. And we want to set that equal to 0. In other words, when p is equal to a half. Why is that interesting? Well, it's interesting because it means that the rate of evolutionary change is maximized when our genetic variance is highest. Our genetic variance is highest when our A allele, our capital A allele, and our little a allele are at the same frequency. That's the maximum rate of change is going to happen when we have the most genetic variance in the population. This is interesting because it turns out more generally, the rate of evolutionary change, the rate of adaptation is maximized when we have the most genetic adaptation in the most genetic variance in the population. So more genetic variance means a greater rate of adaptation. Less genetic variance means evolution can proceed slower. This is a really fundamental result, pops out of a very simple model that is broadly applicable in evolutionary biology. So even these very, very simple models can tell you something very general and profound. More genetic variation means a faster rate of adaptation. Okay, that's it for today. I'll see you guys next Monday.